So it is my pleasure to introduce Dr. Pedro Beltrao, the first speaker of our series. Dr. Beltrao earned his undergraduate degree in biochemistry and biology from Coimbra University in Portugal and his PhD degree at the EMBL PhD program with a fellowship from the Portuguese Foundation for Science and Technology. He spent 4 years as a postdoctoral fellow in the laboratory of Nevan Krogan at UCSF, USA, and held a group leader position at the EMBL EBI in Kingston, UK. Since January 22, so this year, Dr. Beltrao is associate professor at the Institute of Molecular Systems Biology and Department of Biology at the ETH Zurich in Switzerland. Dr. Beltrao's research interest is with evolution of cellular interaction networks, investigating how post-translational regulatory systems have evolved and how they function in present-day species in health and disease. He also investigates genotype to phenotype changes and employs bioinformatics and systems biology approaches in much of his research. More recently, Dr. Beltrao contributed to several high-profile studies on the phosphorylation landscapes of SARS-CoV-2 and the functional landscape of the human phosphoproteome. He also investigated the sequence features mapped onto 3D structures of proteins that define kinase specificity, and he participated in efforts to structurally resolve the human proteome interaction networks using AlphaFold2, the now famous deep learning-based protein structure prediction engine that was developed by Google DeepMind. Today, Dr. Beltrao will tell us about the assessment of large-scale applications of AlphaFold2 in structural bioinformatics. So, Pedro Beltrao, your Zoom floor is for you now.
Thank you for the kind introduction. I'm gonna try to hide these controls a little bit. Yeah, so I hope you can see it. I hope everybody can hear me well.
Yeah,
so
we can see you. Yeah.
So as Shoshana was mentioning, so I'm going to describe some of uses that we're having of AlphaFold2 and predictions from AlphaFold2. And so just first of all, just to give a quick introduction to the themes that we focus on in the lab. So as I was mentioned, we're very interested in this relationship between what happens at the level of DNA and eventually what happens at the level of the organism. And for that, of course, there's a lot of work that is required. It's a problem that we're not going to solve in our lifetimes, but different people study different aspects of this. You can be interested in variation in genetics that leads to changes in proteins and their dynamics and their interactions, but also then at the level of the cells, how these different proteins come together to form molecular cellular functions. And of course, eventually at the level of the organisms and even ecosystems. And so in our group, we try to connect across these different scales of organization by thinking about potential computational transfer systems, transfer functions. So in the sense, if I see genetic variation in the DNA, I would like to, first of all, be able to say where these variations lie. So for example, if they would be within a protein coding region, we should be able to say something about the impact of that mutation at the level of protein stability, interaction, reaction rates, and so on. And then we continue on over to aspects that have to do with organism, which I'm not going to go into today because, of course, this talk today is very much at this level of the atomistic scale. But to give you this flavor that a lot of the work that we do in our group is to try to combine different approaches to go from atomic scale up to organismal fitness level. So, but then, of course, I think everybody here is interested in protein structures, and there's much to learn about protein structures. Sometimes it's good to remind other people that proteins are not just sort of little cartoons in a diagram, but there's a lot to learn from protein structures. And for example, as I just said, we would like to be able to say if I find a missense variant, what's the consequence of that mutation at the level of stability, interactions, binding of small molecules, and so on. So AlphaFold2, I think, and other people I think share this feeling that is a transformative leap forward in the capacity to predict protein structures, which leads to many outcomes that we're going to touch upon today. But of course, it's important to note and to stress that AlphaFold2 doesn't come out of anywhere, not out of nowhere. There was a lot of developments, including the whole process of building a resource where a lot of structures were deposited and so on. But after July, when AlphaFold2 was released, there was a lot of excitement online. So many people started sharing through Twitter many different applications and use cases of this. And I thought it was a really exciting time, and I just reached out to a number of people that were sharing these initial results, and we tried to put together this compendium of use cases that's online. It's in the review, but you can read it in the preprint. And of course, you can see just from the author list, it's an effort that comes from many different people, and many different groups contributed to different aspects, which I'm going to show you here in a second. So some of the things that we wanted to address is, to what extent does AlphaFold give you more relative to other approaches like homology model, and what can we gain in particular from the uncertainty that the method gives us. And I'm sure many people now would have had different introductions to AlphaFold2, but I just want to point out some key things because they're important for what's going to come next. So of course, just as an introduction, you require an input sequence. This input sequence is used to fetch homologous sequences to build a multiple sequence alignment. It's also used to fetch homologous sequences that have already solved structures, so potentially homologous structures. So template searches. It's thought that this template-based search itself is not very important, because if you ablate this, they've done this in the original publication. If you ablate this, in fact, you don't have a very strong reduction in power. What you do get is, the important part here is this multiple sequence alignment. And again, just to make sure everybody's on the same page, for the idea, it's a very old idea, is not from DeepMind's idea that you could have correlated patterns of mutations along the sequence of these homologous proteins across different species that would give you hints that two positions within the protein should be closely linked in space. And that's really the major power behind this approach. And of course, the neural network model, highly complex neural network model that extracts signal out of these multiple sequence alignments. And then again, for simplicity, you get a prediction out at the end. And the important thing is, not only do you get a prediction of what the structure would look like, you also get predictions for uncertainty. And this is really key. Of course, many predictions do not come with uncertainty measurements. So there are two types of uncertainty or error estimates in the model. One error is trying to estimate how the confidence that AlphaFold2 has in modeling any single position, which is this PLDTT score. And so for really high PLDTT scores, it's thought that, above 90 or so, that at the level of side chains, it could be accurate enough. And then another uncertainty, another error measurement is for the relative position for any pair of positions within the protein sequence, which is shown here by this heat map. So we have each position in a matrix. So each pair of position, you have an estimate of how likely that the relative position between these two positions is correct. So what this also means is you could have an estimated high accuracy in a given position, say, for example, a whole region of this protein over here, but then you could still be not very confident about the relative orientation of these subregions of the protein. So this is going to be important for later. And so just the take-home message of a lot of things that I'm going to show you today is that you should really consider using these metrics of uncertainty in whatever process that you then end up using or doing in the follow-up of having one of these structures. So you have one of these predicted structures, and then as structural bioinformaticians, you may do many other things with it, or you could also be an experimental person. So you have to consider these uncertainties in order to follow up your analysis later. So one of the things that you asked, and this is work from Arthur, we wanted to know, to what extent do we gain a lot of new knowledge compared to homology modeling? So homology modeling is already a very high confidence way by which you can derive structural predictions. And so if you just do homology modeling, in this case, what we've done, we took models from Swiss model, which already had predicted structures by homology model for several species. And we compare the coverage that one would get at the level of individual proteins in this plot here, or at the level of the individual residues. And we just ask how much more would you get in terms of predictions from this. And so on average, what we get is something more around 25% of novel predictions that are of high confidence. So if I split the AlphaFold2, which is the gain part that's now modeled, of course, AlphaFold2 will model everything, but then only some part of this will be high confidence. And this is split here into different degrees of confidence. And if you just take above 70 as a reasonable confidence, which are these blue colors here to the right, on average, you gain something like 25% novel predicted residues with high confidence. It's also worth noting that it's not equal to all species. And this could have to do also with the sequence coverage for other species by which they could already have a lot of genome sequencing available. And in other words, what this also does, it transforms a structural prediction problem into a problem of just having more genome sequences for the right species of interest. So let's say if you wanted to, in this particular case for Plasmodium, is an example where you see that there's actually on average, lower confidence in the predictions, and there's less residues that are well modeled in this way. And Plasmodium is one of these cases. It's a very different eukaryotic species, and there's actually not well covered in terms of genome sequencing for other related species. So if one would not I suggest, right, this is an hypothesis that hasn't been shown to be true, but I suggest that if we would then to be able to sequence certain species that are at interesting evolutionary distances away from Plasmodium, you'd be able to then make better structural predictions for this particular species. Okay, so another interesting note that was already found in the AlphaFold paper, but it's because Balint and Norman had a closer look into this, is this idea that if you look again at this example structure on the left, you have these regions that are of low confidence. That are, of course, they are modeled here in one way, but in sense that what the confidence score is also telling you is that the AlphaFold is not confident in any way. So these positions may not have any structure, or they may have some structure, and also the relative orientation, where this is placed, is also not very certain. And so there was this idea that maybe the lack of confidence suggests that these regions are not structured, that the lack of confidence maybe predicts the fact that these will be intrinsically disordered regions. And so Balint and Norman sort of tested this with a benchmark data set. And here's just summarizing this with an area in the ROT curve. Again, 0.5 would be random, 1 would be perfect. And comparing IOPRED, which is a standard disorder predictor that many people would use. It's also very easy to use, even if there will be other ones that are maybe slightly more performant. But if you compare this standard disorder predictor against metrics that you could derive from AlphaFold, such as surface accessibility or windows of surface accessibility, or the individual confidence metric for each position, or windowed versions of that, then you get a performance that outperform the standard disorder predictors. And this is, I think, without, I think there's probably some more sophisticated ways, and I think Norman and Balint are working on more sophisticated ways of doing this. Another thing that I think we only scratched the surface of work by Giannini and Mehmet, but I think is one of the most exciting areas of future research, is the idea that we are going to have these predictor structures for what's going to be likely 100 million protein sequences from the whole kingdom from all kingdoms, so from the whole tree of life. So in this case, at the time where Giannini and Mehmet did this, there was around 300,000 predictor structures from 21 species, and to that they added 100,000 protein structures from the PDB. They've took care to remove some of these regions of low uncertainty because these complicated a bit these type of analysis. But the analysis they were trying to do is to do an all by all structural comparison. And so it becomes non-trivial at this point to do all by all structural comparisons. And so the way they've decided to do this for efficiency's sake as well is to subdivide each individual structure into different substructure elements into an approach that Giannini and Mehmet have developed in the past. So each protein is defined as a vector of subshapes in such a way that they're independent of orientation. And so then they can be compared. In a sense, you design a vector that has counts of individual substructures within the structure, and then you could more easily compare these vectors across all of these protein structures in a way that's more efficient. And so in this representation is a 2D representation. So it's in this case, is a TC representation of the distances between all possible structures. So 2 points that are close together in this representation will be more structurally similar because they are represented by the same structural elements. And they are colored by also their sequence family membership as defined by sequence, so protein from sequence. And what you'd hope to see is that protein sequences from the same protein family should be having the same set of structural elements, and therefore they should be close together in the space of structural element representations. And this is what we get out of this. And of course, based off of this, you can also then define clusters. You can cluster this space of representation and ask whether you find any new clusters that don't have any experimental data, any experimental models assigned to them. So any clustering exercise will, of course, have you can define either bigger or smaller clusters, and there's always some discussion on the best ways to do clustering, and we didn't go too much into this, but we did find several clusters, and these are sort of described in the paper, where there were the majority, so more than 95% of the structures found here were not from the PDP, which would represent structures that are not well populated from experimental data, let's say. We found one example of a potential novel structural element, and I think there's much more work to do in terms of using these approaches to find new folds, new structural elements. And I find that this, in particular from the point of view of evolution, this is going to be, I think, a treasure trove, because this will allow us now to study evolution, so look for deep homology using structural information, and to better understand the evolution of shapes and evolution of function as well. I think an interesting point to make here is really also this difficulty from the point of view of algorithms. So I think for structural bioinformatics, most of all, we were used to dealing with maximum of, let's say, like 100,000 structures. And really this now will need to scale up a lot of our approaches. So now you're going to have 100 million protein structures, and therefore a lot of the algorithms that we're using are not scalable up to that level. Okay, so that was a quick view into some of the overviews, and then we took these predictor structures and we used them on a couple of different attempts of things that we would typically use structures for. So one thing that we do is we took these AlphaFold structures, and then using other software try to predict the impact of a mutation and ask if the AlphaFold structure is as good as an experimental structure. And so the same way we looked at protein pockets, I'll show you a bit of that. I'm not going to show you, but of course a lot of work that's been done as well now is taking these AlphaFold predicted structures together with experimental data and ask if this helps you build models when say the data is not good enough, for example. But the take-home message for all these things is really that you can take into account these uncertainties, and when you take into account the certainty or the error, you get as good as an application as you would get with an experimental structure. Okay, so I'm going to focus now on this idea of the pockets. And this is work done by Jürgen, a postdoc in our group. And if this example structure that I've been showing you, this is a kinase, the EGFR receptor. So what you would typically what you would expect, so there's a transmembrane region. This is a membrane-bound receptor, and you'd see that this transmembrane region should be giving an orientation that's schematized here on the right. So you have this N-terminal region that should be on the outside. You have the cytoplasmatic region, and in principle, these two should never be together, right? It's only an artifact of the fact that AlphaFold doesn't know what a membrane is, and this uncertainty, and it tries to sort of fold these in such a way that it will place these things together. And so if you do a standard approach to find a pocket within this structure, whatever approach you use, you can have these pockets that are clearly wrong because they are formed between regions that shouldn't be together. And the other thing is you have these linker regions, or let's call them low uncertainty in these predicted elements, which could be disorder regions, and these could also either form pockets that shouldn't exist, or form or occlude pockets that are real. Okay, so you're going to look a bit into this, and you took a data set of 300 proteins where you know where the pockets are, because there's structures being solved with and without ligands, so bound and unbound. And he made some pocket predictions and looked at the size distribution of the pockets in the true pockets that are known, in pockets predicted from structures of the bound or unbound form, and then on structures from AlphaFold2 for exactly the same proteins. And what you typically see is that in the case of AlphaFold2, as in this example that I just showed you, you get maybe these larger pockets, so slightly skewed to, this is the size of the pocket, so you get slightly skewed larger pockets, which already hints at the fact that these are not likely to be correct. So on this size here, what you're going to try to do is looking at the mean confidence score for the residues that are assigned to the pocket. And what you see is that the smaller pockets also tend to have higher confidence of the residues at the pocket. And so, in other words, one could potentially use either the size or the confidence metrics to exclude pockets that are likely to be incorrect or driven by this uncertainty. So if you take into account these errors, so the probability that these residues are not correctly modeled, then you have a better overlap with the true pockets. So this is shown here by looking at this direct overlap with the known true pockets, either from the bound structures. On the bound structures, you have a sort of similar performance. So higher here means you have a better overlap. If you take all of the structures from AlphaFold, or if you subdivide by this confidence in the pocket predictions of the residues in the pocket. And so if you do this, you tend to have just a better overlap. So these pockets that are spurious, these pockets that are made up of residues that have like low confidence are more likely to be spurious and do not overlap with the true pockets. Of course, there is a little bit of circularity here, because these are cases where you do have an experimental structure. And so one potential thing that we would be confounding for is the fact that these have lower confidence because they don't overlap with regions for which there could be a template in the PDB. So another test that excludes this confounding issue would be to take protein sequences for which there is no homology whatsoever. So in this case, there is no templates that could be used. And some of these proteins, so about 969 proteins are such human proteins that have no homology. 180 of these have been annotated as enzymes, even though there is no structures for them. And so these 180 being enzymes, they should more likely have pockets because they're enzymes, therefore there should be a pocket to do the catalysis. And so the idea was, can we use pocket prediction to predict whether one of these 969 proteins is an enzyme? And this, again, we do a good job of. So if you take the predicted structures from AlphaFold and we try to find pockets in these proteins, this is the error in the rocker for trying to predict these 180 enzymes. And it does matter a little bit whether you filter the pockets or not. So this is shown here between this blue AUC versus this orange AUC, where you have a slight but significant improvement in prediction power with this. And of course, then once you have this, you could do other types of approaches, such as template matching. This is done by Roman from Janet Thornton's lab, where they've run these template-based matches where you try to find other combinations of residues that have been described to perform certain catalysis or molecule binding, like zinc ion binding and other types of configurations. And by doing this, you can take a sequence that has no homology in the PDB. We make an AlphaFold2 prediction, we find a pocket, and then we use these template searches to suggest what this pocket could be doing, for example. And one interesting thing that we found in this example, but there is many other examples and many other people have been finding similar ideas, is that when you look at the side chains that AlphaFold is predicting, it is predicting in such a way that it looks like the bound form. In this case, so looking at the ion binding residues. And this is not just for this protein. Other people were noticing this. In fact, there was an interesting study from this group here. This is not our study. They were systematically looking at cases where there was a bound and unbound form of the protein, and looking at the residues in the binding sites, and looking at the AlphaFold2 models. And what they find is that for 70% of the cases, these are predictions for the bound state of the protein. Even though AlphaFold does not have the ligands in there, there's no knowledge of the ligands, the predictive structure the AlphaFold2 generates tends to be biased towards the configuration of the bound states, which is an interesting observation. Okay, so an application of this that we've been working on is to take, so in a sense, what this is also telling you now for the whole proteome of human, but any other species, we have these prediction structures, and we have ways of characterizing pockets and at least coming with some confidence metrics to say whether this pocket seems to be reasonable in some sense. So now this opens the door for whole proteome-wide docking exercises, truly open, like a whole proteome now. And there's, of course, many other applications in biotechnology for discovery of enzymes and so on. So what we've been doing in this case, so Jürgen still, is we've been making use of these large compendiums of drug screening data sets where for the same cell lines, there's been drug and CRISPR screens done. So this is done by the Broad and the Sanger. And when from these types of studies, what you can do is you can correlate the essentiality of a given gene in these cell lines against the sensitivity of a drug that you treat the cells with. As this is shown here, each point here is a cell line. And you can see the cases where the cell lines will tend to die if you delete EGFR by a CRISPR screen. And also these same cell lines in this case are quite sensitive to this particular drug. In this scenario, you could suggest that this drug, one of the mechanisms could be that it's inhibiting EGFR. So that's a hypothesis that you generate out of these types of screens. So by doing, there's different distance metrics that one can get out of this. A simple one would be correlation, of course. So you can correlate the drug response to a given drug against all CRISPR screens, so all genes in the CRISPR screen. And the ones towards the top would be candidates for being the drug targets of that drug. And so if you do this in a very global sense, you get reasonably good prediction power for known drug targets based off of this. And so we thought that this would be a good opportunity as well to use this idea of being able to do whole proteome structural docking using AlphaFold2 predictive structures. And the way we're trying to do this is we first take the CRISPR screen associations that I just showed you for a given drug, like lapatinib. We rank the associations from the genetics data sets. And then we do docking on the top ranked associations. The idea being that for the associations from the genetics, from the genomics data sets, you don't know if there's a direct or an indirect effect of the drug. It could be that the drug is binding a protein, and then it could have a secondary consequences through the action of that inhibition. And that's why some of these other genes would also appear to be essential in the same cell lines. And so some of these then we could re-rank based off of the docking scores to be able to say which of these that are genetically associated are also likely to have a drug binding pocket that would be a good drug pocket for that protein, for that drug. And in this case, this re-ranking is actually effective. So the two drug targets of this drug are EGFR and RBB2. These are two known drug targets. The other proteins here are much less likely to actually be true binders of the drug. And then when you re-rank based on the docking score, you do get some of these get lower ranked based off the docking, which makes sense. So these two things together should in principle be useful. And if we do this a bit more systematically, again, if you just take the genomics rank, which is really a very good signal, you can get a slight improvement, at least, for when you combine this genomics-based ranking together with a structural-based ranking. And so, say for time's sake, I'm going to sort of skip this and go just to this protein complex prediction part of it. And so, Shoshana was saying, so many people right at the beginning of AlphaFold noticed that although AlphaFold2 was trained to predict the structures of a given individual protein, there were people on Twitter showing examples where if you just, instead of giving one single protein sequence, what if I give AlphaFold 2 protein sequences that I know should interact, and I just put a linker connecting them. And they noticed immediately that AlphaFold2 could come up with a prediction for the complex, even though it was thinking that was an individual protein, of course. Which is, of course, exciting. Predicting individual structures is exciting, but predicting structures of protein complexes for the combinatorial reason of it, is even more exciting because it's so much harder to get structures for large complexes. And it's a really unintended application. AlphaFold2 was not trained to do this. And in this initial compendium collection of assessments we did, there was a series of tests for the prediction of either oligomerization, heterooligomerization, and also some predictions with the domain disorder interaction. And there, I should say, there are other groups, of course, that have published preprints with other similar ideas and similar tests. So Arne Ellefsen, a person that's been doing really a lot of work on this area of using AlphaFold2 for the prediction of protein complexes, he provided a benchmark data set. So he provided a test with a benchmark set that's commonly used for standard docking applications, and which is shown here. So each protein here is a pair. So each dot here is a pair that's you're trying to predict the structures of. And on one axis, you have a prediction score based off the AlphaFold2 structure prediction for the complex. And then the other one is using a standard docking software method. Of course, for the docking method, you require already having the structures of the individual monomers before you even try to dock them. So, of course, keep in mind that AlphaFold2 already does the individual protein structure prediction as well. But you get a really vastly superior outcome by trying to simultaneously predict the structures of the complex together using AlphaFold2. Here is the DocQ score, which a little sort of is a bit like IMSB, but very much focused on the interface residues. So if it's higher than this 0.23, you can say that you have a somewhat accurate prediction, at least of the interface positions. So about 30% of these AlphaFold2 predictions were deemed to be accurate versus 7% if you were using a standard docking approach. And you can also come up with scores that are based off of the interface residues, both the interface size and the confidence of the mean confidence of the interface residues themselves. And so after this initial ideas, there were improvements made. So Arne's group tried to improve the way the alignments are done. And this was boosting in their performance tests to about 60% of the models being correctly predicted. And DeepMind itself came out with a new parameterized model. So the neural network itself was retrained to try to predict structures of protein complexes. And in this paper, they've claimed on the order about 60% correct for structural prediction protein complexes. The initial version of this had some problems. In fact, there was a lot of the vast majority of the proteins that you'd predict using AlphaFold monomer were coming out with clashes, really serious clashes. Sometimes more than 20–30% of the protein was clashing between one protein and the other protein. In the last, I think it was 3 weeks ago or so, they've re-updated their model, and now these updated versions don't have these clashes, which I'm surprised that they didn't see it, because I guess they just didn't open a few of those proteins to see that there were serious problems. So one big important point is all these assessments are done for cases because you are comparing them with the PDB structures for protein complexes. These are all cases where you already know that the two proteins physically interact and they form an interface. So for all these cases, then you get at least 60% correct prediction of the structure. If you blindly predict whether two proteins, if you try to blindly predict the structure for a complex for any random two proteins, of course, AlphaFold will still make a prediction, but you wouldn't know that these form a direct interface. And so for these 60% are not actually the correct assumption of accuracy for just a random blind prediction of two proteins. So together with Arne, then we were using this idea to increase the scale. So let's just predict protein structures of complexes for many human protein interactions. So how far could we potentially take this? And we took a data set of 65,000 human protein interactions that are known to interact. And 55,000 of these were from yeast to hybrid assays, 10,000 were from pull downs. And just to make sure everybody is more or less familiar with these ideas. So the yeast to hybrid assay tends to find protein interactions that are likely to be that direct interactors, in the fact that they should share an interface. But it also tends to find things that are more transient, so low stability, potentially smaller interfaces. So the pull down assays, on the other hand, are more likely to find things that have high binding affinities and should maybe more likely have bigger interface sizes. But they could also find cases where it's not a direct interaction, because you pull down a protein, there may be 3 proteins coming attached with it. And for example, one of these may not form a direct interface, but could be an indirect interaction. So when you make these predictions, we can come up with a confidence metric, which is this predicted DocQ score, which is a combination of the interface size plus the average confidence of the model at the interface residues. And Arne's group developed this, and there's a good correspondence between this predicted DocQ score and the experimentally determined DocQ score. And what we could see here is, if you just look at the random distribution, which is this blue, so this is a random set of protein pairs that we try to predict, you get confidence scores, this estimate that we get very close to 0. If you look at cases where there's a known crystal structure, which is the red ones, they tend to be highly shifted to these high predicted, high confidence scores. So if you look at the yeast to hybrid, which is this dotted line, you get a much worse performance in terms of this confidence metric. And if you look at the pull down data set, which is this black dashed line, you tend to have a higher confidence in average. And so without sort of going too much into this, one take home from this type of looking at different types of interactions, what it looks like is you just tend to have higher confidence of the prediction and also more accurate predictions for cases where the interaction that you were trying to predict before you tried the prediction, these are were likely to be high binding affinity, large interfaces, and with information that it's direct interactions. So in any case, based off these confidence metrics, we can estimate that we get something on the round of 3,000 to 9,000 high confidence predictions. So most of these, around 80% of these, are likely to be true, to be correct. So an alternative way that we had to assess whether our predictions were correct was using publicly available mass spectrometry data that tells you about cross-linking information. So by cross-link MS, you can find 2 regions in the protein across the 2 proteins in the structure that are linked by the cross-link. So they should be at a distance that's determined by the size of the cross-link. In this case, all the cross-links that we use should be less than 30 angstroms in distance, which is still a fairly reasonably large distance there. But nevertheless, this allows us to ask if, for all the pairs for which we have cross-linking data, how many of these cross-links are within this 30 angstroms limitation that they should be in. And so for about 50% of the cases, we have at least one cross-link that's within the required 30 angstrom distance. And if we split the predictions based off these pre-DocQ score, then we can get up to 75% predicted structures that have at least one cross-link at the required distance. So this is similar to the types of numbers that we're getting in the comparisons with the PDB. And so just to go back to the beginning, of course, why would this be interesting? Once we have these prediction structures, we can say a lot about mechanisms of either protein mutations or protein post-translation modification. So this, and in the paper, we describe examples of this type of application. And so finally, one last thing is all these predictions that we were making are for pairs of proteins. So just two proteins, one at a time. But many of these complexes exist in much larger conformations. And so one way that we can try to build these larger complexes is by superimposition. So we can take each pair, superimpose a common subunit, and build up a bigger complex. And so this is what we're showing you here for this RFC complex, where we overlap several of these pairs by superimposition of one of the common subunits. And in this case, the gray at the end is the cryo-EM structure. And you can see that you get a fairly decent outcome of the whole complex. There are some areas that are not well predicted, but there are many areas that look actually quite well predicted. Of course, the problem is that there are many combinations of pairs. So there's going to be many potential outcomes of these predictions. And there will be many potential predicted substructures along the way, right? So then it becomes a bit complicated, and there's an algorithmic challenge. So for the computational method development approach, there is the idea, of course, that you can now develop methods to try to combine these predictions together in a larger assemblies. And again, just for time, I'm going to show you an illustration, though, of something if you go towards the end of this type of idea of assembling larger complexes by a superimposition of common subunits, you can get slightly different conformations. So in this case, here in the middle, we have two predicted conformations that depend on how you assemble the subunits together. And in this case, it is actually corresponding to true differences. So in gray here on the right, I have the overlay with this open conformation of this TF2H core complex. And on the left is an overlay of the closed conformation. And in this case, you can see that this subunit here towards the bottom does open and close relative to the rest. And it sort of corresponds fairly well with this open and close conformations that have been experimentally determined. Okay, so then Arne's group already published another preprint where they show one way by which you can do this type of search across the possible configurations, and it's also some metrics to try to quantify the potential error in this type of assembly. So you can, but again, I think there's going to be a lot of exciting challenges on trying to do this in different ways. Okay, so just a summary of some things that I've said. So you can get confidence predictions for probably on average more than 25% of extra residues that you couldn't get before. There is this computational challenge. We're going to have 100 million structures, and many of the approaches that we were used to using do not scale to 100 million. So we have to think of new ways of doing this. And of course, a lot of novel biology. I think evolution in particular was going to be an exciting time to look at evolution, and also from biotechnology, so new enzymes and things like this. The take-home message again, just make sure that you use this uncertainty in the models, and there should be better, clever ways of taking this uncertainty into your applications. And you can get reasonably good prediction of protein complexes, which again opens a lot of interesting applications. Okay, what it doesn't do, it predicts structure, but it doesn't tell you much about folding. I can discuss this. There's already some ideas that some empirical evidence that it's learned biophysics. It doesn't inform us on protein dynamics, although you can try to bias in such a way that will try to predict different conformations. It doesn't predict the structure of mutated proteins. Again, I can discuss this more. And there are sort of no predictions for binding to small molecules, DNA, RNA, and so on. I would like to take this opportunity just to say that we have the two positions for structural bioinformatics, so maybe some in the audience could be interested in. So this is a large collaboration between the Institute for the Gulbenkian Science in Lisbon, UCSF, and ETH Zurich. There's 2 positions of 5 years funding that will be based in Lisbon, but many opportunities to spend periods of time in San Francisco or Zurich. And with that, I will stop and address any questions.
Thank you so much for this large, big overview of all this interesting work of the community and yourself. So that's really impressive. Now we have opened the question and answers period. I don't have many questions, but I'm sure they will come as we go along. So there is one first question is a bit longish, but I'll read it. Does the inclusion of known structures in the calculations induce a bias in the outcome of the prediction, especially in the case where there is strong local conformational heterogeneity between members of a protein family? And there he gives an examples, for example, the CDRs of antibodies that are incorrectly predicted or when predicting complexes. I noticed, for example, that prediction of complexes with nanobodies often fail completely. So the nanobodies and the antibodies are in special case. So please comment on this.
Yeah, I guess these antibodies are special. So I'm not sure the question. So does the inclusion of known structures and calculation induce bias in the outcome of prediction? So I think, again, if you go back to even the original publication, if you ablate the template search in the prediction, you get almost no decrease in accuracy. So I don't think there's, at least from the point of view of the template search part of the prediction, but of course there's a lot of potential biases that have to do with the training itself. So not the fact that it went and searched for a template, but the fact that it was trained on what we know, right? So and so that, I think there's a lot of open questions on what has AlphaFold learned and how much biased it is to the fold space and to the specific elements that are being solved to date. So that's as much of like, if anybody wants to chime in.
Yeah, I would just add to say that the antibodies are really special because of their evolutionary, history. In other words, it's hard to get multiple sequence alignments that would be, that you can exploit for that. So that's why they are, they are a special case. Okay, there's another question is, are all multiple sequence alignments equally good for AlphaFold predictions? Have you evaluated the properties of an ideal MSA to generate a good prediction?
We haven't. Sergey has done a bit of work on this. I guess the question is more, how much do you need? And that's really one of the strengths of AlphaFold2 is it does a lot more with lots fewer sequences in that multiple sequence alignment. And again, this has to do, back to this idea that AlphaFold2 has learned biophysics. It's not just a good way of extracting information from the multiple sequence alignment. And Sergey has done a really cool study where they've looked at, no multiple sequence information and just providing AlphaFold2 with different starting templates that are different conformations of the same protein, where the lowest energy form of the protein. And without any multiple sequence alignment information, the outcome is that the AlphaFold2 will score each of the initial proteins from the ensemble, each of the initial structures, and it scores most confident the lowest energy form, the true lowest energy form. So that tells you that there's something there that's by physics, not just a good way of extracting information from the multiple sequence alignment.
But I think he also says that the multiple sequence alignment helps the procedure to sample.
That's I think an intuition.
Okay.
As far as I can tell. So that
I was also wondering what it meant.
I think that's the intuition, is that of course you need the multiple sequence alignment
to sample.
Yeah, but that's other people have sort of noted this. In fact, now we have this object, which is this neural network that people are trying to figure out, because of course maybe there's something quite interesting about how folding. We want to understand folding, and so this neural network to some extent has some knowledge, but we don't actually are able to pull it out of it. So it's an object of study in itself.
I think so. It's going to be really interesting. So there is another question here. How could you tell more about how could you tell more about how AlphaFold tries to handle dynamics in proteins? Should they improve their predictor to include these dynamics, or should a whole new predictor specific for dynamic proteins be developed?
Yeah, so it doesn't, there's nothing in it for dynamics, really nothing. So it's really not meant for anything to do with dynamics. The only thing is that there's some people who have tried to bias the predictions by, again, providing different templates. So forcing the templates that are given and so on, or just changing the number of times that it recycles and these types of things. And that will lead you to different protein predictions, slightly different predictions towards the end. And then there's some hints that these differences could be sort of linked to dynamics. But I really think you need something else. I think it's not meant for that. And it's related to the point mutation, so that maybe there's a question next to it. Then so
okay, yes, you comment more on single point mutation predictions, which is also linked to the dynamics. Yeah.
Exactly. So again, it's linked to this dynamic. And so in our hands, when we tried cases where there were two conformations that depend on a mutation, it was not a mutation because we were looking at phosphosides, but it's more or less the same
similar,
thing. Yeah. So you have the experimental structure of the different forms, so modified or not modified, and we were trying to predict that difference. And what happens is it will always end up in the same structure, same fold, same conformation. Sometimes it will be the modified, sometimes it will be the not modified, but for a given sequence, it's highly sort of skewed to give you one outcome. So if there's few changes, even though they will probably, even when they experimentally are known to lead to a different conformation, it will still just give you one conformation of that sequence with few modifications. And so again, I guess this is again tied to this notion that in reality, a sequence is not a given structure, but it's a dynamic. There's different conformations and there's different energies, right? So some are lower energy, some are higher energy. And AlphaFold2 is not made to give you the different possible solutions and rank you by energy. It just gives you one, which is what AlphaFold thinks is the lowest energy form, right?
But this brings me to another question very quickly. In a number of cases, it has been shown, for example, in the prediction of single conformations, you get the bound conformation, actually the conformation that is bound to another protein. I'm not talking about bound to a ligand, but mostly to another protein. For example, only the open conformation, you have a protein, it has an open conformation in the PDB and a closed conformation, and the open conformation is the only one that's predicted. And it's in a number of cases. And this, so what is there, what is it due to? Is that, what the actually evolutionary speaking in the cell, you have that. The most common conformation is that which is bound. And this is what comes, the information that is provided by the multiple sequence alignments, or what, how do you explain this?
Yeah, so my and I'll get to that point, but my view is what I was describing is that imagine that there's multiple conformations, and so AlphaFold doesn't really tell you that they exist and where are the energy differences. But I was always having this mind that you would always tend to predict the lowest energy form, but the bound state is often not, right? Which exactly, so that's a case where it's not the answer to that. Which then I don't.
Maybe the most biologically, from the sequence from the multiple sequence alignments, there's some information is entering the procedure that gives you the biologically meaningful state. Anyway, we cannot answer this easily. Okay, there is one other question is in screening. Okay, how many new folds would you expect to discover once you have, all this plethora of structures, 100 million structures in your hands?
I don't know. No, but I think re-emphasizing here, I think this is really, I think it's going to be a fantastic treasure trove for biology, right? I think there's a lot of research to do there, but I don't know the answer.
Well, where would the most new structures come from? Not new folds, but the additional structures come from what kind of species? Do you have?
Oh, the coverage is going to be essentially any essentially all new sequences, because it's going to cover, once you reduce the redundancy and the protein universe by sequence redundancies, so removal, they're going to go through all of it.
Mm-hmm. Mostly microbes, you think?
Yeah, that's essentially, it's going to go for microorganisms. Yeah.
Okay. And now another question: in screening with one protein for potential receptors, what are the best parameters to evaluate interactions in predicted complexes, and how to separate the correct from the not correct predictions of interactions? Yes.
Yeah, so this predicted DocQ score is described in our RNS papers, and it's a combination of, so they've tried different things. We also played around a bit with that. If you look at the size of the interfaces, how many residues at the interface, and the average confidence, average predicted confidence from the AlphaFold prediction for the residues at the interface.
Mm-hmm.
But this bias is towards large interfaces because it's in the score. So yeah, that's what's currently working best, but please feel free to try other things.
Yeah, the most transient complexes will be harder to predict for sure.
Yeah, but this is in fact partially reality, right? But yeah, maybe it requires special attention.
Mm-hmm. Maybe just one remark, when you showed the data from the comparison of the complexes predicted by AlphaFold2 and docking, you basically said, you mentioned GRAM, one docking program, which is GRAM, and it's definitely not the standard one. I mean, not the best one. There are many other ones. So I think one should be careful in just when presenting this type of results, saying that it was tested against, I know it was tested against other programs as well. And I'm sure that, I feel that the prediction, the performance will be really much more general, for really, for many complexes from AlphaFold2, but one has to be careful.
Yep, no, I take the point.
Yeah. Okay. Another one is from Anastasia. What is your take on the future of handling big experimental designed and predicted protein structure data sets? What would be the challenge according to you?
Oh,
in other words, the beacon program and the beacon data set. Anyway.
The challenge is just scale. So any operation that one does, if you're doing at sort of 10,000 versus 100 million, just from the purely computational, very basic sort of things, like you want to compare 100 million structures all by all, is absolutely non-trivial. It's not something that you could just easily run on a cluster, even if you have a big cluster. So you have to actually change the algorithms now for many of these things. So come up with sort of clever solutions to these problems.
Yeah. More data, more difficult to
analyze
everything and get something out of it.
Order of magnitude is different. So even basic things like clustering, all of these things become you have to do clever sort of approximations to the problem and just see whether you still get accurate enough results.
Yeah. Okay. This is more or less the last question. I have one more concerning your building the three-dimensional structures using AlphaFold2 on the networks, on the PPI networks. And it's actually quite striking that the better results you get with AlphaFold2 for the APMS, for the complexes, basically from the networks that are derived from affinity purification mass spectrometry rather than yeast-to hybrid. Have you tried to see, use co-expression data to try to filter out, we have done this in the past comparing yeast-to-hybrid data to APMS, and yeast-to-hybrid was abysmal. It kind of the co-expression data for yeast-to-hybrid are random. So I think that this makes you really wonder what the yeast-to-hybrid really tell you, whether it's really only transient complexes or non-specific interactions in general, because their expression profile, the expression of the proteins are high level, mostly, and not in the normal context. So what is your take on this?
Yeah, I mean, yeah, I truly don't think that Easter hybrid is sort of all crap. Let's be it.
Well,
yeah,
why? I mean, yeah.
Yeah, as you're saying, I think, you know, the biological explanation is that it captures a lot of these low stability, low binding affinity directions, and maybe these are also not as highly coordinated in expression because they don't need to be as highly coordinated in expression.
That's the nice political answer, and I think it's the true, you know, it's the true dilemma.
Yeah, so there's of course,
dilemma.
and then of course it could be just a much higher error, right? Obviously, right?
Yeah,
so but regardless of that, is really this question of, you know, a lot of things inside the cell bump against each other, and some of these are
sometimes
functional.
It's useful, and sometimes, you know, you don't know what's doing.
Some are functional, some are not functional, some are productive and required, and some are not. And so if whatever it is from our ideas, how do we tell these apart, right? And this is quite challenging. And so it's always easier to look at these nice, large, stable protein complexes, both computationally and experimentally. And so we are very interested in signaling. So for us, you know, looking at kinase signaling, for example, is something that we care about. And again, all the predictions and all the experimental stuff is much harder. So we take it is, of course, we need to take whatever we can. But I also should say, from this idea of using AlphaFold, in particular, because the scoring system is biased by interface size, right? So also by that nature, it will be not very positive towards the East2 hybrid if biologically it is like real interactions that happen to have lower surfaces.
Yeah.
Yeah. So then it's, you know, it's always this problem. It's probably a biological explanation and also technical issues.
So, Pedro, in your lab, do you use experiments and computation, or what is your plan?
Yeah, so we now for a couple of years, it's about 20% experimental work that we do, but that's all on the yeast genetics side, and now we're also going to do some aspect.
And are you going to use AI-based deep learning methods in your approaches? Can you say a word or two about this? Because this is, you know, the area that we would like to, you know, put forward and develop, you know, at the VIB and the Structural Biology Center.
Yeah, so we do have one where we had a project on neural network development, specifically neural network development, and that was a variant effect predictor. But I think we were a bit too ambitious. So we were trying to predict variant effects from sequence alone, not using multiple sequence alignments, and we don't get better than state-of-the-art things. Deborah Marks at a very similar. They had a really nice paper trying to do exactly the same thing, but using multiple sequence alignments. So the advantage that we have with our method, it's not published yet, but we're going to put a preprint out relatively soon, is that it is extremely scalable because we don't use multiple sequence alignments. It's just purely from sequence. So there's no step of sequence search or multiple sequence alignment building, anything. So then it can run through hundreds of millions of protein sequences in really fast timescales. But it's not better than the other things. So it doesn't help for a human, for example, but trying to do variant effect prediction in bacteria, or if you want to do it at a really large scale, it's useful.
Okay, thank you. So no more questions, and thank you very much for this interesting and motivating webinar, the first webinar in the series.